Your brain weighs about 3 pounds. It contains roughly 86 billion neurons. Each neuron connects to thousands of others, creating a network so complex that mapping it remains one of science's greatest challenges. For decades, scientists believed that consciousness emerges from the electrical signals firing between these neurons, like a biological computer processing information through circuits of nerve cells. This classical view treats the brain as an incredibly sophisticated machine, where consciousness is simply the result of billions of neurons talking to each other through chemical and electrical messages. But what if this entire framework is wrong? What if the true source of human consciousness operates at a scale so small and through mechanisms so strange that we have been looking in the wrong place all along? A groundbreaking new study has detected ultraviolet superradiance from tryptophan networks, and this discovery could completely reshape our understanding of what makes us conscious. Superradiance is not just ordinary light emission. It is a collective quantum phenomenon where molecules arranged in precise crystal-like structures emit light together in perfect synchronization, producing an intense burst of energy that far exceeds what individual molecules could create alone. Think of it like this. Imagine a stadium filled with people clapping randomly versus everyone clapping in perfect unison. The synchronized clapping creates a powerful, coherent sound wave. Superradiance works the same way at the molecular level, but with light instead of sound. The fact that this quantum behavior has been observed in tryptophan networks is extraordinary, because tryptophan is an amino acid found abundantly in the structural proteins of your brain, particularly inside tiny tube-shaped structures called microtubules that exist within every neuron. This discovery matters because it provides the first experimental evidence that the biological structures in your brain might actually support quantum effects, something most scientists thought was completely impossible in the warm, wet, noisy environment of living tissue. For context, quantum effects typically require extreme conditions like temperatures near absolute zero and complete isolation from environmental disturbances. Your brain operates at 98.6 degrees Fahrenheit. It is filled with water and ions constantly moving around, and it experiences constant electrical noise from neural activity. These conditions should destroy quantum states almost instantly, which is precisely why the scientific community largely dismissed the idea that quantum mechanics could play any role in brain function or consciousness. Yet here we are, with experimental data showing that under the right circumstances, brain molecules can exhibit quantum behavior. This finding directly connects to one of the most controversial and fascinating ideas in modern science: Roger Penrose's quantum consciousness theory. So Roger Penrose is not just any scientist making wild speculations. He is a Nobel Prize-winning mathematical physicist from Oxford University who made groundbreaking contributions to our understanding of black holes, working alongside Stephen Hawking. Physicist Lee Smolin called him one of the very few people who, without reservation, qualifies as a genius. In 1989, Penrose published a book called The Emperor's New Mind, where he proposed something radical. Human consciousness cannot be explained by classical physics or traditional neuroscience alone. He argued that consciousness is non-algorithmic, meaning it cannot be replicated by any computer program, no matter how sophisticated. According to Penrose, there is something fundamentally different about conscious experience that goes beyond mere computation. His solution to this mystery involved quantum mechanics, the branch of physics that governs the behavior of particles at the smallest scales, where the normal rules of reality break down and particles can exist in multiple states simultaneously until they are observed or measured. Penrose suggested that consciousness arises from quantum processes happening inside the brain, specifically through the collapse of quantum superpositions. A quantum superposition is when a particle exists in multiple states at once, like the famous thought experiment of Schrödinger's cat being both alive and dead until you open the box. Penrose believed that neurons could maintain quantum superpositions and that consciousness emerges when these superpositions collapse into definite states. He even proposed that gravity itself plays a role in causing these collapses, creating discrete moments of conscious awareness. This was an extraordinary claim that most neuroscientists and physicists rejected outright, because it seemed to violate everything we knew about how quantum mechanics works in biological systems. The skepticism was understandable. Quantum coherence, the delicate state required for particles to remain in superposition, is notoriously fragile. In laboratory experiments with isolated particles at near-zero temperatures, quantum states can be maintained for brief periods. But in the brain, with its warm temperature, constant molecular motion, and endless sources of interference, calculations suggested that any quantum superposition would collapse in about 10 to the power of negative 13 seconds. That is far too fast to have any meaningful impact on brain processes, which operate on time scales of milliseconds to seconds. This seemingly insurmountable problem led the vast majority of scientists to dismiss quantum consciousness theories as interesting philosophy but terrible science. The criticism intensified when philosophers and physicists pointed out that Penrose provided no concrete mechanism for how neurons could possibly maintain quantum states long enough to matter, and no experimental evidence supported his ideas. For more than 3 decades, quantum consciousness remained on the fringe of mainstream science, considered by most researchers to be a beautiful idea with no basis in physical reality. However, science advances through unexpected discoveries, and the new evidence of ultraviolet superradiance in tryptophan networks represents the first real crack in the wall of skepticism. If brain molecules can indeed exhibit quantum behavior under certain conditions, then perhaps Penrose was not as far off as everyone thought. Perhaps consciousness does have a quantum component, operating through mechanisms we are only beginning to understand. This possibility has profound implications not just for neuroscience and our understanding of the mind, but for fundamental questions about the nature of reality, free will, and what it truly means to be conscious. The journey to understanding whether quantum mechanics really does play a role in human consciousness starts with understanding Penrose's original theory in detail, examining why it was dismissed, and then exploring the new experimental evidence that is forcing scientists to reconsider what they thought they knew about the brain. That journey begins with a deeper look at what Penrose actually proposed and why his ideas challenged the very foundations of both neuroscience and physics. The human brain performs approximately 1 exaflop of operations per second, which equals 1 quintillion calculations. That is a 1 followed by 18 zeros worth of computational power happening inside your skull right now. Most neuroscientists believe this massive computational activity, carried out by billions of neurons exchanging electrical and chemical signals, is sufficient to explain everything about consciousness. They argue that awareness, emotions, thoughts, and even your sense of self are nothing more than the emergent properties of this biological computer. But Roger Penrose fundamentally disagreed with this computational view of consciousness, and his reasons for rejecting it form the foundation of one of the most provocative scientific theories ever proposed. Penrose's central claim was that consciousness is non-algorithmic, meaning it cannot be reduced to a series of step-by-step computational processes that a machine could replicate. His argument was not based on mysticism or vague intuition. Instead, it emerged from rigorous mathematical logic, specifically from examining what humans can do that computers provably cannot. In his 1989 book, The Emperor's New Mind, Penrose used Gödel's incompleteness theorems to argue that mathematical understanding involves insights that go beyond any algorithmic process. Gödel proved that in any consistent mathematical system, there exist true statements that cannot be proven within that system using its own rules. Human mathematicians, however, can recognize these truths through understanding and insight, something that transcends mechanical computation. Penrose argued that if consciousness were purely computational, we would be limited by the same constraints that bind computers, but our ability to grasp mathematical truths suggests something fundamentally different is happening in our brains. This led him to propose that consciousness must involve some non-computational physical process, and the only place in physics where non-computational elements exist is in quantum mechanics, specifically in the mysterious process called quantum state collapse. To understand Penrose's theory, you need to grasp what quantum superposition actually means. In classical physics, objects exist in definite states. A coin is either heads or tails, a light switch is either on or off, an electron is in one location or another. But in quantum mechanics, particles can exist in multiple states simultaneously until they are measured or observed. This is the famous principle illustrated by Schrödinger's cat, the thought experiment, where a cat in a sealed box is both alive and dead at the same time, until someone opens the box and observes it. Penrose proposed that neurons in the brain could similarly contain quantum superpositions existing in multiple states at once. According to his theory, these quantum states are not just random noise, but are organized in ways that influence neural processing. The critical moment for consciousness happens when these quantum superpositions collapse into definite states. Standard quantum mechanics describes this collapse as happening when a measurement occurs, but what counts as a measurement and why collapse happens at all remain deeply mysterious questions that physicists have debated for nearly a century. Penrose offered a radical solution. He proposed that collapse is not caused by measurement or observation, but is instead an objective physical process driven by gravity itself. This idea is called objective reduction, and it represents a genuine modification to standard quantum mechanics, rather than just an interpretation of existing theory. The gravitational collapse mechanism works like this. When a particle exists in a quantum superposition of being in two different locations, Each location has a slightly different gravitational field because mass curves space-time according to Einstein's general theory of relativity. This means the superposition creates two slightly different space-time geometries, one for each possible position of the particle. Penrose argued that space-time itself cannot remain in superposition indefinitely because the energy associated with maintaining these different geometries becomes unstable. When the energy difference reaches a certain threshold, the superposition spontaneously collapses into one definite state, and this collapse is a real, physical event, not just an updating of our knowledge about the system. The time it takes for collapse to occur depends on the mass involved. Larger masses create bigger differences in space-time curvature, and therefore collapse faster, while smaller masses can maintain superposition longer. This gravitational orchestration of quantum collapse Penrose proposed is what generates moments of conscious awareness. Each collapse event produces what he called a moment of proto-consciousness. And when billions of these events are organized and orchestrated by brain structures, they give rise to our continuous stream of conscious experience. This means that consciousness is not something that causes quantum collapse by observing it, as some interpretations of quantum mechanics suggest. Instead, consciousness is caused by quantum collapse through an objective gravitational process that happens regardless of whether anyone is watching. Penrose's proposal was extraordinary for multiple reasons. First, it connected two of the biggest unsolved problems in science, the nature of consciousness and the measurement problem in quantum mechanics. Second, it suggested that our current theories of physics are incomplete and need modification to account for how quantum mechanics and gravity interact at small scales. Third, it implied that consciousness has a fundamentally different character than computation, meaning that artificial intelligence systems based on classical computing could never truly become conscious no matter how sophisticated they become. A computer processes information through algorithms, following predetermined rules to transform inputs into outputs. But if consciousness involves the non-computational collapse of quantum states orchestrated by gravity, then it represents something genuinely beyond what any algorithmic system can achieve. This has profound implications for questions about machine consciousness, free will, and the nature of understanding itself. Penrose was essentially saying that when you understand a mathematical proof or have a creative insight or experience the redness of red, something is happening in your brain that no computer simulation could ever replicate, because it involves physical processes that transcend computation. The scientific community's response to Penrose's theory was overwhelmingly skeptical, and the criticism came from multiple directions. Neuroscientists pointed out that there was no evidence whatsoever that neurons operate using quantum mechanics. Brain imaging studies, electrophysiology experiments, and detailed models of neural circuits all seem to work perfectly well using classical physics. If quantum effects were essential for consciousness, you would expect to see some signature of quantum behavior in neural activity, but none had ever been detected. Physicists raised a different objection. Quantum coherence is incredibly fragile and requires extreme isolation from environmental disturbance. In physics laboratories, maintaining quantum superpositions requires cooling systems down to temperatures near absolute zero, which is minus 273.15 degrees Celsius, and shielding them from any vibrations, electromagnetic interference, or thermal fluctuations. The brain operates at 37 degrees Celsius, is filled with water molecules constantly jostling around, contains ions flowing through membranes, and experiences continuous electrical activity from neural firing. These conditions should cause decoherence, the destruction of quantum superpositions, almost instantaneously. Calculations suggested that any quantum state in the brain would collapse in about 10 to the power of negative 13 seconds due to environmental noise alone, long before gravity could have any effect and far too fast to influence neural processes that operate on millisecond timescales. This decoherence problem seemed to completely rule out any possibility of quantum mechanics playing a role in brain function. Philosophers also challenged Penrose's arguments. Many disputed his use of Gödel's theorems to prove that consciousness is non-computational, arguing that his logical reasoning contained subtle flaws and that Gödel's results do not necessarily imply anything about human consciousness. Others questioned whether understanding really requires non-computational processes, or if Penrose was making an unjustified leap from mathematical insights to claims about physics. Additionally, even if consciousness does involve quantum mechanics, it was not clear why this would make it non-computational, since quantum computers are still computational devices that process information through algorithms, just using quantum bits instead of classical bits. The theory also faced the challenge that it made no specific testable predictions about brain function. Penrose could not point to any particular experiment that would confirm or refute his ideas, because he had not identified which structures in the brain might maintain quantum coherence, or how these quantum states would interface with classical neural activity. Without a concrete mechanism or experimental predictions, the theory remained in the realm of speculation rather than testable science. For more than 2 decades after Penrose first proposed his quantum consciousness theory, it remained on the fringes of respectable science, discussed more in philosophy departments and popular science books than in neuroscience laboratories. Most researchers considered it an interesting thought experiment that almost certainly had nothing to do with how the brain actually works. The consensus was that consciousness must emerge from classical neural computation, and that invoking quantum mechanics was an unnecessary and implausible complication. However, science has a way of surprising us, and what seemed like an impossible idea was about to gain an unexpected ally and a concrete biological mechanism that would transform quantum consciousness from abstract speculation into a testable hypothesis. Absolute zero is minus 273.15 degrees Celsius, or 0 Kelvin, on the absolute temperature scale. At this temperature, all molecular motion theoretically stops, and particles reach their lowest possible energy state. In physics laboratories around the world, scientists use sophisticated cooling systems to bring quantum computers and experimental apparatus down to temperatures within a few thousandths of a degree above absolute zero. They do this because quantum coherence, the delicate state where particles maintain quantum superposition, is extremely fragile and requires near-perfect isolation from environmental disturbances. The slightest thermal vibration, stray electromagnetic field, or interaction with other particles can destroy quantum states through a process called decoherence, where the quantum system becomes entangled with its environment and loses its special quantum properties. This need for extreme cold is not just a technical limitation that better engineering could overcome. It reflects a fundamental challenge posed by the laws of thermodynamics and quantum mechanics. Heat is molecular motion. When you heat something up, you are making its atoms and molecules move faster, vibrate more intensely, and collide more frequently. Each of these movements and collisions represents an interaction that can disturb a quantum state. The hotter the environment, the more violent these disturbances become, and the faster quantum coherence disappears. This creates what seems like an insurmountable problem for any theory suggesting that quantum mechanics plays a role in brain function, because the human brain operates at approximately 37 degrees Celsius, which is 310 Kelvin, incredibly far from the near absolute zero conditions that quantum systems normally require. To understand just how hostile the brain environment is to quantum states, consider what is actually happening inside your skull right now. Your brain is about 73% water, with the rest consisting of fats, proteins, salts, and other molecules all packed into a dense active tissue. Water molecules are constantly moving, rotating, and colliding with each other thousands of billions of times per second. Dissolved ions like sodium, potassium, calcium, and chloride are flowing through channels in cell membranes, creating electrical currents and changing charge distributions throughout the tissue. Neurons are firing action potentials, generating electromagnetic fields that propagate through the surrounding medium. Metabolic processes are breaking down glucose and oxygen to produce energy, releasing heat and creating concentration gradients that drive further molecular motion. Every single one of these processes creates noise and disturbance that can interact with and destroy quantum superpositions. The brain is not just warm, it is wet, noisy, and constantly changing, creating what physicists would call a highly decohering environment that seems fundamentally incompatible with maintaining quantum states. The theoretical calculations paint an even bleaker picture for quantum consciousness theories. In the year 2000, physicist Max Tegmark published a famous paper calculating how long quantum superpositions could possibly survive in brain structures like microtubules. His analysis considered the rate at which environmental interactions would cause decoherence, taking into account the temperature of brain tissue, the density of water molecules, and the electromagnetic environment created by neural activity. The result was striking. Tegmark calculated that quantum coherence in microtubules would last only about 10 to the power of negative 13 seconds. To put this timescale in perspective, light travels about 30 nanometers in that amount of time, which is roughly the width of a large protein molecule. Neural processes, by contrast, operate on timescales of milliseconds to seconds. An action potential, the electrical signal that travels down a neuron, lasts about 1 millisecond, which is 1,000th of a second. Synaptic transmission, where neurotransmitters cross the gap between neurons, takes several milliseconds. Cognitive processes like recognizing a face or understanding a word require tens to hundreds of milliseconds. If quantum states are collapsing in 10 to the power of negative 13 seconds, they would decay and reform trillions of times during even the fastest neural event. This makes it seem impossible for quantum effects to have any meaningful influence on brain function, let alone form the basis of consciousness itself. The decoherence problem goes even deeper when you consider the specific requirements for quantum computation. For quantum effects to be useful in processing information, you need not just isolated quantum superpositions, but also quantum entanglement between multiple particles and the ability to perform controlled operations on these quantum states before decoherence destroys them. Quantum computers achieve this by operating at temperatures below 1 Kelvin, carefully isolating their qubits from vibrations and electromagnetic interference, and performing calculations in microseconds before decoherence sets in. Even with all these precautions, maintaining quantum coherence long enough to perform useful calculations remains one of the biggest engineering challenges in quantum computing. The idea that biological tissue at body temperature could somehow maintain quantum coherence, not just for microseconds, but for milliseconds or longer, and do so reliably enough to form the basis of consciousness, seemed to violate everything physicists knew about how quantum systems behave in warm, noisy environments. This is why most scientists dismiss Penrose's theory immediately. It was not that they disliked the idea philosophically, or thought quantum mechanics was inherently incompatible with biology. Rather, the physics appeared to make it impossible, and without a concrete mechanism to explain how the brain could overcome the decoherence problem, there seemed no point in pursuing the theory further. Critics also pointed out that quantum effects generally become important only at very small scales or very low energies, neither of which seemed relevant to brain function. Individual atoms and electrons behave quantum mechanically, certainly, but when billions of them come together to form molecules and cells and tissues, their quantum behaviors typically average out into classical physics. This process is called self-averaging, and it is why macroscopic objects like brains, chairs, and buildings obey Newton's laws, rather than requiring quantum mechanical descriptions. There are exceptions, of course. Superconductors maintain quantum coherence across macroscopic distances, but only at extremely low temperatures. Quantum tunneling allows particles to pass through barriers they classically could not overcome, and this does occur in some biological processes like enzyme catalysis. But these are isolated quantum events that happen at specific molecular sites, not sustained quantum computations spread across large networks. For quantum mechanics to play the kind of central role in consciousness that Penrose proposed, you would need something truly extraordinary, a biological mechanism that could maintain quantum coherence across many molecules for physiologically relevant timescales, despite operating at body temperature in a wet, noisy environment. No known physical system could do this, and there seemed no reason to believe that evolution would have discovered a way to do it when classical neural computation appeared perfectly adequate for explaining brain function. The temperature problem also raised questions about evolutionary plausibility. If consciousness required quantum effects, and quantum effects required low temperatures, how could warm-blooded animals like mammals and birds be conscious? Did consciousness disappear if you had a fever? Did cold-blooded animals like reptiles and fish have different kind of consciousness depending on water temperature? These questions seemed absurd, suggesting that the whole framework of quantum consciousness was misguided. Evolution builds on physical mechanisms that are robust and reliable under the conditions where organisms actually live. A consciousness mechanism that was exquisitely sensitive to temperature and vulnerable to thermal noise would be evolutionarily disadvantageous compared to classical neural processing that works reliably across a range of body temperatures and environmental conditions. The fact that brains function effectively as biological computers using electrical and chemical signaling suggested that this classical mechanism is what natural selection actually discovered and refined over hundreds of millions of years. Adding quantum mechanics on top seemed unnecessary and implausible, like suggesting that mechanical clocks secretly rely on nuclear physics to keep time. While technically everything is quantum mechanical at the deepest level, invoking quantum effects to explain macroscopic phenomena requires demonstrating that those effects actually matter at the scale and conditions of interest, and for the brain, all the evidence suggested they did not matter at all. This scientific consensus, supported by detailed calculations and fundamental principles of physics, relegated quantum consciousness theories to the realm of interesting speculation rather than serious neuroscience. For Penrose's ideas to gain traction, something dramatic would need to change. Either the physics would need to be wrong, which seemed unlikely given how well-tested quantum mechanics is, or someone would need to discover a specific biological mechanism that could somehow overcome the seemingly impossible challenge of maintaining quantum coherence in the warm, wet chaos of living brain tissue. Part 4. Stuart Hameroff's edition, Microtubules. Xenon is element number 54 on the periodic table, a noble gas that makes up approximately 0.09 parts per million of Earth's atmosphere. It is colorless, odorless, and chemically inert, meaning it does not react with other substances under normal conditions. Despite this chemical inertness, xenon has a remarkable property. When you breathe it at certain concentrations, it causes complete unconsciousness. This is not just sleepiness or sedation, but a total loss of conscious awareness, which is why xenon is used as an anesthetic in some medical procedures. The puzzle that fascinated Stuart Hameroff, an anesthesiologist at the University of Arizona, was this. How can a gas that does not chemically react with anything in the body cause such a profound change in consciousness? Traditional pharmacology explains drug effects through chemical reactions. A drug molecule binds to a receptor protein, changing its shape and triggering a cascade of biochemical events that alter cell function. But xenon does not bind to receptors in the conventional sense because it does not form chemical bonds. Other inert gases like argon, krypton, and even nitrogen under high pressure can also cause anesthesia, and they share xenon's property of being chemically unreactive. This suggested to Hameroff that anesthetics might not work by interfering with chemical signaling between neurons, but instead by disrupting something more subtle and fundamental about how brain cells maintain consciousness. Hameroff began investigating this mystery in the 1980s, focusing on a class of cellular structures called microtubules. Microtubules are tiny, hollow tubes found inside virtually every cell in your body, including neurons. They are made of a protein called tubulin, which assembles into cylindrical structures about 25 nanometers in diameter. To put that scale in perspective, A nanometer is 1/1000th of a meter, and a human hair is roughly 80,000 to 100,000 nanometers wide, so microtubules are thousands of times smaller than the width of a hair. Despite their microscopic size, microtubules play crucial roles in cellular function. They form the structural skeleton that gives cells their shape. They act as highways along which motor proteins transport cargo from one part of the cell to another, and they are essential for cell division, pulling chromosomes apart when cells replicate. In neurons specifically, microtubules run like rails along the length of axons and dendrites, maintaining the cell's elaborate branching structure and facilitating the transport of neurotransmitters, receptors, and other molecules needed for neural signaling. Most neuroscientists viewed microtubules as purely structural support elements, important for cell biology, but irrelevant to information processing and consciousness, which were thought to happen entirely through electrical signals traveling between neurons at synapses. Hameroff saw something different when he looked at microtubules. He noticed that they possess a highly ordered crystal-like structure. Each microtubule is composed of 13 parallel strands called protofilaments, and each protofilament is a linear chain of tubulin protein subunits stacked end to end. The tubulin proteins are arranged in a precise geometric lattice, creating a regular repeating pattern similar to atoms in a crystal. This structural regularity intrigued Hameroff because it suggested the possibility of coherent vibrations and information processing happening within the microtubule lattice itself. Furthermore, tubulin proteins contain pockets of aromatic amino acids, particularly tryptophan, which is the largest naturally occurring amino acid and has unusual electronic properties due to its complex ring structure. Tryptophan contains an indole group with delocalized electrons that can absorb and emit light, particularly in the ultraviolet range. Hameroff hypothesized that these tryptophan residues within the ordered microtubule structure might be able to interact through quantum mechanical effects, potentially creating coherent electronic states that could process information in ways completely different from classical neural computation. The connection to anesthesia became clearer when Hameroff examined where anesthetic molecules actually go in the brain. Studies using radioactive tracers and other techniques showed that general anesthetics, including xenon and other noble gases, accumulate in hydrophobic pockets within proteins, which are water-repelling regions formed by nonpolar amino acids clustered together. The tubulin proteins in microtubules have exactly these kinds of hydrophobic pockets, and experiments demonstrated that anesthetic molecules bind preferentially to these sites. Hameroff proposed that when anesthetic molecules occupy these hydrophobic pockets in tubulin, they subtly alter the protein's shape and electronic properties, disrupting whatever quantum mechanical processes might be occurring in the microtubule lattice. This would explain why chemically inert gases cause unconsciousness. They are not triggering chemical reactions, but are instead physically interfering with quantum states in microtubules through their mere presence in specific binding sites. The fact that anesthetics affect single-celled organisms like paramecia, which have no neurons but do have microtubules, supported this idea. When you expose paramecia to anesthetic gases, they stop swimming and become immobile, suggesting that the anesthetic is disrupting fundamental cellular processes, mediated by microtubules rather than specifically targeting neural synapses. Hameroff's proposal was revolutionary because it shifted attention from neurons to subcellular structures as the potential site of consciousness. In the standard neuroscience model, consciousness emerges from networks of neurons firing action potentials and releasing neurotransmitters at synapses. Information processing happens at the level of neural circuits, with individual neurons acting as the basic computational units. But if Hameroff was right, then the real action might be happening inside neurons, within the microtubule networks that pervade the cell. Each neuron contains millions of tubulin proteins organized into thousands of microtubules, creating an internal computational substrate that is orders of magnitude more complex than the neuron itself. If microtubules could process information, then the brain's computational capacity would be vastly greater than anyone had imagined, and consciousness might arise from quantum processes happening at the nanoscale within these intracellular structures, rather than from classical electrical signals passing between cells. The timing of Hameroff's work was crucial because it coincided with Roger Penrose's search for a biological mechanism that could support his quantum consciousness theory. Penrose had proposed that consciousness requires quantum effects, but had not identified where in the brain these effects might occur or how they could be protected from decoherence. When Hameroff contacted Penrose in the early 1990s and described his ideas about microtubules, Penrose immediately recognized that this could be the missing piece. Microtubules had several properties that made them potentially suitable for maintaining quantum states, despite the challenging brain environment. Their crystal-like structure meant that vibrations and electronic excitations could potentially spread coherently across many tubulin subunits, rather than being localized to individual molecules. The ordered geometry created a kind of shielding effect where the interior of the microtubule tube might be partially protected from the surrounding cellular chaos. The hydrophobic pockets where electrons were likely to spend time were relatively isolated from water molecules, reducing one major source of decoherence. And the sheer abundance of microtubules in neurons meant there was enormous potential for quantum information processing if the mechanism could be made to work. Penrose and Hameroff joined forces to develop what they called the Orchestrated Objective Reduction Model, or ORCH, OR, which combined Penrose's gravitational collapse mechanism with Hameroff's microtubule substrate. The basic idea was that tubulin proteins in microtubules could exist in quantum superpositions of different conformational states, meaning the protein could be in multiple, slightly different shapes simultaneously. These superpositions could become entangled across many tubulins, creating macroscopic quantum states involving thousands or millions of proteins. According to Penrose's objective reduction mechanism, these quantum states would spontaneously collapse due to gravitational effects when the mass-energy difference between superposed states reached a critical threshold. The collapse would occur after a time interval determined by the number of tubulins involved and the mass-energy difference, with larger superpositions collapsing faster. When collapse occurred, the quantum state would reduce to one definite classical configuration, and this moment of reduction would be accompanied by a moment of conscious experience or proto-consciousness. The orchestration part referred to the idea that these quantum collapses are not random, but are organized by classical neural activity and molecular processes in ways that create meaningful conscious content rather than just noise. Together, quantum superposition in microtubules and gravitationally orchestrated collapse created a complete model linking subcellular quantum mechanics to human consciousness through a specific biological mechanism. This transformed Penrose's abstract theory into a testable hypothesis because it made specific predictions about where to look for quantum effects and what would happen if you disrupted them with anesthetics or other interventions. Every cell in your body contains a complex internal skeleton called the cytoskeleton, which is not made of bone but rather of protein filaments that crisscross the cellular interior like scaffolding. The cytoskeleton has three main types of filaments: microfilaments made of actin protein, intermediate filaments made of various proteins depending on cell type, and microtubules made of tubulin protein. Among these, microtubules are the largest and most rigid, functioning as the structural girders that maintain cell shape and organization. In neurons, microtubules are particularly abundant and play essential roles beyond simple structure. They form long parallel arrays that run the entire length of axons, which can extend over a meter in large animals like giraffes or whales. These microtubule highways are used by motor proteins called kinesins and dynins, which walk along the microtubule surface carrying cargo such as neurotransmitter vesicles, mitochondria, proteins, and other cellular components from the neuron cell body to distant synaptic terminals and back. Without this microtubule-based transport system, neurons could not function because diffusion alone is far too slow to move materials across such long distances. During cell division, microtubules form the mitotic spindle that pulls duplicated chromosomes apart, ensuring each daughter cell receives the correct genetic information. In developing neurons, microtubules guide the growth of axons and dendrites, helping these processes navigate through the brain to find their correct targets and form appropriate connections. The structure of microtubules is remarkably precise and geometrically elegant. Each microtubule is a hollow cylinder approximately 25 nanometers in outer diameter and 15 nanometers in inner diameter. The cylinder wall is composed of 13 parallel columns called protofilaments arranged in a ring. Each protofilament is a linear polymer of tubulin dimers stacked end to end, with each dimer consisting of two closely related protein subunits called alpha tubulin and beta tubulin. The alpha and beta tubulins alternate along each protofilament, creating a polar structure with a distinct plus end and minus end. The 13 protofilaments are arranged so that the tubulin subunits form a precise helical lattice when you look at the microtubule surface, with each tubulin surrounded by 6 neighbors in a hexagonal pattern. This crystalline arrangement means that microtubules have long-range structural order similar to atomic crystals like diamond or silicon, but made of much larger protein molecules. The regularity of this structure allows physical properties like mechanical vibrations, electronic excitations, or quantum states to potentially propagate coherently across many tubulin subunits, rather than remaining localized to individual molecules. Stuart Hameroff proposed that this crystal-like lattice structure makes microtubules ideal candidates for quantum information processing. His reasoning was based on several key features of tubulin and microtubule organization. First, each tubulin protein can exist in at least two distinct conformational states, meaning the protein can adopt different three-dimensional shapes by rotating parts of its structure. These conformational changes involve movements of electron clouds within the protein, particularly in hydrophobic pockets containing aromatic amino acids like tryptophan, phenylalanine, and tyrosine. Second, because tubulins are packed closely together in the microtubule lattice, with only about 8 nanometers separating the centers of neighboring dimers, their electron clouds could potentially overlap and interact through quantum mechanical effects like tunneling and resonance. Third, the tubulins are connected not just within each protofilament, but also laterally between adjacent protofilaments through specific binding interfaces that could allow conformational states to propagate across the entire microtubule surface. Fourth, the interior of the microtubule cylinder is relatively isolated from the surrounding cellular water, potentially providing some protection from decoherence. And fifth, microtubules are not static structures, but are dynamically regulated by proteins called microtubule-associated proteins, or MAPs, which bind to the microtubule surface and can influence tubulin conformation and the overall stability and dynamics of the polymer. Hameroff hypothesized that microtubules could support quantum computations through what he called quantum coherent oscillations in the tubulin lattice. The basic idea is that tubulins enter quantum superpositions of different conformational states, and these superpositions become entangled across multiple tubulins through their interactions in the lattice. An entangled state is a quantum state of multiple particles where the particles are correlated in ways that cannot be explained by classical physics. For example, if two tubulins are entangled, measuring the conformational state of one instantly determines the state of the other, even though neither had a definite state before measurement. When many tubulins become entangled, they form a collective quantum state that spans a significant portion of the microtubule, or potentially across multiple microtubules connected through their associated proteins. This quantum state could in principle perform quantum computations, processing information through quantum interference and superposition in ways that classical systems cannot. The hydrophobic pockets within tubulin, where electrons are likely to spend time, could act as qubits, the quantum bits that are the basic units of quantum information. These electron clouds could tunnel between different locations or energy states, creating quantum superpositions that encode information. The orchestration aspect of the theory, which gives orchestrated objective reduction its name, refers to how these quantum processes are organized and controlled to produce meaningful consciousness rather than random noise. Hameroff proposed that microtubule-associated proteins and other cellular regulatory mechanisms orchestrate the quantum states by determining when and where quantum coherence develops in the microtubule networks. Classical neural activity, such as action potentials and synaptic inputs arriving at the neuron, could influence microtubules through various mechanisms including calcium, ion influx, phosphorylation of microtubule-associated proteins, and changes in membrane potential that affect the electric field surrounding microtubules. These classical influences would prime certain regions of the microtubule network to develop quantum coherence while suppressing it in other regions. The quantum states would then evolve according to the Schrödinger equation, exploring multiple computational paths simultaneously through quantum superposition. When the quantum state reached a critical threshold determined by the gravitational self-energy of the superposition, it would undergo objective reduction as described by Penrose's theory, collapsing to one definite classical state. This collapse event would constitute a moment of conscious experience, and the particular state selected during collapse would be influenced by non-computable factors embedded in the geometry of space-time at the Planck scale. The Orch-Orr model made several specific quantitative predictions. First, it predicted that the timescale of quantum coherence in microtubules would be in the range of milliseconds to tens of milliseconds, which corresponds to the gamma frequency oscillations observed in EEG recordings at around 40 Hz. These gamma oscillations are associated with conscious perception and attention in neuroscience experiments, suggesting a possible connection to the quantum collapse events proposed by Orch-Orr. Second, the theory predicted that anesthetics would cause unconsciousness by binding to the hydrophobic pockets in tubulin and disrupting quantum coherence, which could be tested by examining how anesthetics affect microtubule properties at the molecular level. Third, the model suggested that consciousness should be affected by manipulations that disrupt microtubules, such as drugs that depolymerize or stabilize microtubules, and that organisms without microtubules should not be conscious. Fourth, if quantum entanglement extends across multiple neurons through gap junctions or other connections, then altering gap junction function should affect consciousness. These predictions provided concrete ways to test the theory experimentally, transforming it from abstract speculation into a scientific hypothesis that could potentially be confirmed or refuted through careful experiments. The model also addressed several philosophical questions about the nature of consciousness. By grounding consciousness in objective physical events, specifically the gravitational collapse of quantum superpositions, Orch-Orr avoided the problems of dualism, where consciousness is treated as something separate from physical reality. At the same time, by incorporating Penrose's non-computable objective reduction, the theory preserved the intuition that consciousness involves something more than algorithmic information processing. The theory suggested that consciousness has a fundamental connection to the basic structure of reality at the Planck scale, where quantum mechanics and gravity meet. This gave consciousness a kind of cosmic significance, linking human awareness to the deepest levels of physical law. The theory also provided a potential explanation for the unity of conscious experience, the fact that you experience yourself as a single unified subject rather than a collection of separate processes. If quantum entanglement can spread across large networks of microtubules throughout the brain, then the resulting quantum state would be a single, indivisible whole, and its collapse would produce a unified moment of experience rather than fragmented pieces. Despite these attractive features and the detailed biological mechanism proposed by Hameroff, the Orch-Orr theory faced immediate and sustained criticism from multiple directions, with skeptics arguing that the physics was implausible, the biology was wrong, and the philosophy was confused. Criticisms that would need to be addressed through experimental evidence before the theory could gain acceptance in mainstream neuroscience. The orchestrated objective reduction model, published by Penrose and Hameroff in 1996, rests on three core principles that together explain how quantum processes in microtubules could generate conscious experience. Understanding these principles requires recognizing how radically different this model is from standard neuroscience, which treats consciousness as an emergent property of billions of neurons communicating through electrical and chemical signals. The OR model instead proposes that the real action happens inside neurons at the level of protein structures too small to see with ordinary microscopes, operating according to quantum mechanical laws that seem to have nothing to do with everyday brain function. The first principle is that microtubules generate quantum states due to their crystalline structure. This claim is the foundation of everything else in the theory, and it directly contradicts the conventional wisdom that quantum effects cannot survive in the warm, wet environment of living cells. Hameroff argued that the regular geometric arrangement of tubulin proteins in the microtubule lattice creates conditions favorable for quantum coherence despite the challenging environment. The tubulins are packed closely together, with their electron clouds nearly touching, separated by distances of only 8 to 10 nanometers. At these scales, quantum mechanical effects like electron tunneling and resonance can occur, where electrons move between neighboring tubulins, not by classical diffusion, but by quantum mechanical processes that allow particles to traverse barriers they classically could not cross. The hydrophobic pockets within tubulin proteins, which contain aromatic amino acids like tryptophan with their extended electron clouds, serve as the actual quantum bits or qubits in this model. These electron clouds can exist in quantum superpositions of different spatial locations or energy states, and when multiple tubulins interact, their electron states can become entangled, creating a collective quantum state that spans many proteins across the microtubule network. The crystal-like structure of microtubules is crucial because it allows quantum states to spread coherently rather than remaining isolated on individual molecules. Think of how waves propagate through a crystal lattice in solid-state physics. The regular arrangement of atoms allows vibrations to travel as collective excitations called phonons that involve many atoms moving together in coordinated patterns. Similarly, in microtubules, the regular arrangement of tubulins could allow electronic excitations or conformational changes to propagate as coherent waves across the lattice. This coherent propagation would help protect the quantum state from decoherence, because random environmental disturbances affecting individual tubulins would be averaged out across the collective state, much like how a large boat is less affected by small waves than a tiny raft. The hollow interior of the microtubule cylinder provides additional protection by creating a partially shielded region where water molecules and ions are less abundant than in the surrounding cytoplasm. Furthermore, the tubulin proteins themselves undergo constant conformational changes as part of their normal function, and these conformational dynamics could be orchestrated to create windows of time during which quantum coherence develops before environmental decoherence destroys it. The key insight is that quantum states do not need to survive indefinitely. They only need to last long enough for quantum computation to occur and for objective reduction to happen, which according to Penrose's calculations could be anywhere from milliseconds to tens of milliseconds, depending on the number of tubulins involved. The second principle of OR is that anesthetics disrupt these quantum states, thereby causing unconsciousness. This prediction flows directly from the mechanism of how anesthetics are proposed to work in the model. General anesthetics are a chemically diverse group of substances ranging from noble gases like xenon to complex organic molecules like propofol and isoflurane, yet they all produce the same effect: complete loss of consciousness at specific concentrations. The classical neuroscience explanation is that anesthetics enhance inhibitory neurotransmitter receptors like GABA receptors, thereby suppressing neural activity and preventing the widespread brain communication thought to be necessary for consciousness. However, this explanation has problems. Some anesthetics do not work through GABA receptors, and blocking GABA receptors does not reliably reverse anesthesia. Moreover, the correlation between anesthetic potency and how well anesthetics dissolve in lipids, known as the Meyer-Overton correlation, suggests that anesthetics work by accumulating in hydrophobic regions of proteins, rather than by binding to specific receptor sites. Hameroff proposed that the relevant hydrophobic regions are the pockets within tubulin proteins in microtubules. When anesthetic molecules enter these pockets, they physically displace electrons and subtly alter the protein's conformational dynamics, disrupting the quantum states that would otherwise form in the electron clouds. This disruption of quantum coherence in microtubules prevents the orchestrated objective reduction events that generate conscious moments, resulting in unconsciousness even though neurons continue firing action potentials and synapses continue releasing neurotransmitters. Recent experimental evidence has strongly supported this second principle. Studies using fluorescence techniques showed that anesthetics directly interact with microtubules, and that this interaction correlates with anesthetic potency better than interactions with any other cellular target. Computational modeling of how anesthetic molecules bind to tubulin predicted the Meyer-Overton correlation, meaning that the theory could reproduce the empirical relationship between lipid solubility and anesthetic potency by assuming microtubules are the primary target. Even more striking, experiments demonstrated that anesthetics dampen quantum optical effects in microtubules, providing direct evidence that anesthetics disrupt quantum processes in these structures. The fact that anesthetics affect single-celled organisms like paramecia, which have microtubules but no neurons, further supports the idea that microtubules rather than neural circuits are the critical substrate. When you expose a paramecium to anesthetic gases, it stops swimming and becomes immobile, then resumes normal activity when the anesthetic is removed. This suggests that even simple organisms have some form of proto-consciousness mediated by microtubules, and that anesthetics disrupt this by interfering with quantum states in the microtubule networks that control cellular behavior. The third principle of OR is that gravity collapses quantum states, producing conscious moments through Penrose's objective reduction mechanism. This is the most radical and controversial aspect of the theory, because it proposes a genuine modification to quantum mechanics rather than just applying standard quantum theory to a new biological context. In standard quantum mechanics, a system evolves according to the Schrödinger equation, which is completely deterministic and describes how quantum superpositions change over time. The superposition only collapses when a measurement occurs, but what constitutes a measurement and why collapse happens remain deeply mysterious. Penrose proposed that collapse is not caused by measurement, but is instead an objective physical process driven by the incompatibility between quantum superposition and general relativity. When a particle exists in a superposition of being in two different locations, each location has a slightly different gravitational field because mass curves space-time. This creates two slightly different space-time geometries, and Penrose argued that maintaining this superposition of geometries requires energy proportional to the gravitational self-energy of the mass difference. When the energy reaches the Planck energy divided by the time the superposition has existed, the superposition becomes unstable and spontaneously collapses to one definite state. The time until collapse is inversely proportional to the mass-energy difference. Larger mass differences collapse faster, while smaller differences can maintain superposition longer. In the context of microtubules, objective reduction occurs when enough tubulins become entangled in a quantum superposition that their collective mass-energy difference reaches the threshold for gravitational collapse. Penrose calculated that for a superposition involving 10 to the power of 9 tubulins, roughly 1 billion protein subunits, the collapse time would be approximately 25 milliseconds. This matches remarkably well with the frequency of gamma oscillations observed in EEG recordings, which are brainwaves around 40 Hz or 40 cycles per second, corresponding to periods of 25 milliseconds. Gamma oscillations are strongly associated with conscious perception in neuroscience experiments. They appear when subjects consciously perceive a stimulus, but not when the same stimulus is presented unconsciously. This temporal correspondence suggests that gamma oscillations might be the macroscopic signature of orchestrated objective reduction events happening in microtubule networks throughout the brain. Each collapse event would constitute a discrete moment of proto-consciousness, and the continuous stream of conscious experience we subjectively feel would be composed of these discrete moments occurring 40 times per second, too fast for us to notice the gaps between them, similar to how movies appear continuous, even though they are actually sequences of still frames projected at 24 or more frames per second. The orchestration aspect, which gives the theory its full name, refers to how classical neural processes organize and guide the quantum states to produce meaningful conscious content rather than random experiences. Microtubule quantum states are not isolated from the rest of cellular function, but are influenced by microtubule-associated proteins, calcium ions, phosphorylation states, membrane potentials, and other classical variables that change based on the neuron's activity. These classical influences select which regions of the microtubule network develop quantum coherence and determine how the quantum states are initialized before they evolve according to quantum mechanics. The quantum computation phase explores multiple possibilities simultaneously through superposition, and the objective reduction collapse selects one outcome that feeds back into classical neural processing, influencing action potentials, synaptic plasticity, and ultimately behavior. This creates a hierarchical system where classical neural circuits orchestrate quantum computations in microtubules, and quantum collapse events in turn influence classical neural dynamics, forming a feedback loop that integrates quantum and classical processes into unified brain function. The enormous potential of this combined classical quantum architecture, if it actually exists, would vastly exceed what either classical or quantum computation alone could achieve. Scientific theories live or die by experimental evidence, not by how elegant or philosophically satisfying they might be. For more than 2 decades after Penrose and Hameroff published the orchestrated objective reduction model in 1996, the theory remained in scientific limbo, neither definitively proven nor completely refuted, existing instead as an intriguing speculation that most neuroscientists dismissed while a small group of researchers defended it. The problem was that testing the theory required measuring quantum effects in living biological tissue, which posed enormous technical challenges. Quantum states are notoriously difficult to detect, even in carefully controlled laboratory experiments with isolated particles at near-zero temperatures. Trying to detect them in the chaotic environment of brain tissue seemed nearly impossible with existing technology. However, as experimental techniques advanced, particularly in quantum optics, nuclear magnetic resonance, and ultra-sensitive detection methods, it became feasible to design experiments that could directly test specific predictions of the OR model. The first wave of these experiments focused on Penrose's objective reduction mechanism itself, attempting to determine whether gravity actually causes quantum state collapse as the theory proposed. If this fundamental physical mechanism did not exist, then the entire OR framework would collapse regardless of whether microtubules could maintain quantum coherence. In 2022, a team of physicists conducted a landmark experiment deep underground in a facility designed to shield delicate measurements from cosmic rays and environmental radiation. The experiment aimed to test whether gravitational interactions could cause spontaneous collapse of quantum superpositions, as Penrose had proposed. The researchers created quantum superpositions of massive particles and carefully monitored how long these superpositions survived under different conditions. According to Penrose's objective reduction theory, larger mass differences in superpositions should collapse faster due to the incompatibility of different space-time geometries, and this collapse should occur even in the absence of any environmental decoherence or measurement. The experiment was designed to distinguish between collapse caused by environmental interactions, which is the standard explanation in quantum mechanics, and collapse caused by gravitational self-energy, which is what Penrose proposed. The researchers used state-of-the-art techniques to isolate their quantum systems from environmental disturbances as much as possible, cooling them to extremely low temperatures and shielding them from vibrations, electromagnetic fields, and stray particles. They then measured the decoherence times for superpositions involving different mass scales to see if there was a signature of gravitational collapse beyond what environmental decoherence alone could explain. The results were disappointing for supporters of OR. The experiment found no evidence of gravitationally induced quantum collapse. The quantum superpositions decayed at rates consistent with environmental decoherence alone, with no additional collapse mechanism related to mass or gravity. This negative result seriously challenged the objective reduction component of Penrose's theory, suggesting that gravity does not cause quantum state collapse in the way Penrose had proposed. For many physicists, this was confirmation that the whole quantum consciousness framework was built on shaky foundations. If the fundamental mechanism that supposedly generates conscious moments does not actually exist, then the entire edifice collapses. Critics argued that the 2022 experiment effectively refuted Core by demonstrating that its core physical mechanism, gravitational objective reduction, has no experimental support. However, supporters of the theory pointed out several important caveats. First, the experiment tested gravitational collapse at mass scales and conditions very different from what occurs in biological microtubules. So it was not a direct test of the OR model itself, but rather a test of the general principle of objective reduction. Second, there are modified versions of objective reduction that do not rely solely on gravity, but might involve other mechanisms for spontaneous collapse. So the failure to detect gravitational collapse does not necessarily rule out all forms of objective state reduction. Third, and most importantly, the experiment said nothing about whether microtubules can maintain quantum coherence or whether quantum processes in microtubules play any role in consciousness, which are separate questions from whether objective reduction occurs. This last point led researchers to shift their focus away from testing Penrose's gravitational collapse mechanism and toward testing the more fundamental and directly testable claim that microtubules can support quantum effects in biological conditions. Even if objective reduction turned out to be wrong or required modification, if microtubules could be shown to maintain quantum coherence and exhibit quantum behaviors like superposition and entanglement, this would still be revolutionary for neuroscience and would suggest that quantum processes might play some role in brain function, even if the specific OR mechanism needed to be revised. This shift represented a strategic retreat to more defensible scientific ground. Rather than defending every aspect of Penrose and Hameroff's original theory, researchers began focusing on the question that could be answered experimentally. Can biological microtubules exhibit quantum phenomena? This question could be addressed through direct experiments on isolated microtubules and on microtubules in living cells, using techniques from quantum biology, spectroscopy, and biophysics. If the answer was yes, it would validate at least part of the OR framework and open up entirely new directions for consciousness research. If the answer was no, it would definitively rule out quantum explanations for consciousness based on microtubules and force researchers to look elsewhere or abandon quantum consciousness theories altogether. The experimental approach required developing new methods to probe quantum properties of biological molecules under physiologically relevant conditions. Traditional quantum mechanics experiments use techniques like interferometry, where particles are sent through double slits to create interference patterns that reveal quantum superposition, or measurements of entanglement through Bell inequality tests that demonstrate correlations stronger than classical physics allows. But these methods are difficult to apply to large protein structures in aqueous solution at body temperature. researchers instead turned to spectroscopic techniques that could detect signatures of quantum coherence in electronic and vibrational states of molecules. One particularly promising approach involved using ultraviolet light to excite electrons in aromatic amino acids like tryptophan, which are abundant in tubulin proteins, and then measuring how the excited states decay back to the ground state. If the molecules are behaving classically, each molecule's excitation and decay would be independent, and the emitted light would have random phases. But if quantum coherence exists across multiple molecules, they could undergo collective emission phenomena like superradiance, where many molecules emit light together in phase, producing a much brighter and faster pulse than classical emission. Detecting superradiance would be direct evidence of quantum coherence spanning multiple molecules in the biological structure. Another experimental avenue involved examining how microtubules respond to anesthetics at the molecular level. The OR model makes specific predictions. Anesthetics should bind to hydrophobic pockets in tubulin, and this binding should disrupt quantum optical effects in the microtubules. These predictions could be tested by comparing the optical properties of microtubules with and without anesthetics present, looking for changes that indicate disruption of quantum coherence. Additionally, the binding sites of anesthetics in tubulin could be mapped using computational chemistry and confirmed with structural biology techniques like X-ray crystallography or cryo-electron microscopy. If anesthetics bind where the theory predicts, and if this binding correlates with changes in quantum optical properties, it would strongly support the microtubule-based consciousness hypothesis, even if the objective reduction mechanism remained unproven. These experiments represented a crucial transition from theoretical speculation to empirical investigation, moving quantum consciousness from philosophy into experimental science. The stage was set for a series of studies that would either validate or refute the biological plausibility of quantum effects in brain microtubules, regardless of what happened with the gravitational collapse mechanism. Fluorescence is the property of certain molecules to absorb light at one wavelength and then emit light at a longer wavelength after a brief delay, typically measured in nanoseconds to microseconds. This phenomenon occurs when photons excite electrons in a molecule to higher energy states, and then the electrons relax back to lower energy states by releasing energy as light. Fluorescent spectroscopy is a powerful tool in molecular biology because it allows researchers to probe the electronic structure and dynamics of proteins and other biomolecules in solution under physiologically relevant conditions. In 2023, researchers used advanced fluorescence techniques to investigate whether anesthetics directly interact with microtubules and whether this interaction affects quantum optical properties of the tubulin proteins. The experimental design was elegant. They isolated microtubules from cells and made them fluorescent by exciting the tryptophan residues embedded within the tubulin proteins using ultraviolet light. Tryptophan is naturally fluorescent because its indole ring structure contains delocalized electrons that can be excited by UV light around 280 nanometers and emit fluorescence around 350 nanometers. By monitoring how this fluorescence intensity changes over time, researchers could study the electronic dynamics of tryptophan in its native protein environment within the ordered microtubule lattice. The key experiment involved comparing fluorescence decay rates in microtubules with and without anesthetics present. Fluorescence decay refers to how quickly the emitted light intensity drops after the exciting UV light is turned off. In classical systems where molecules emit light independently, the decay follows a simple exponential curve determined by the intrinsic properties of the fluorescent molecule. But if quantum coherence exists among multiple tryptophan residues, their electronic states could be coupled, leading to altered decay kinetics. The researchers measured these decay curves with extraordinary precision using time-resolved fluorescence spectroscopy, which can track intensity changes on timescales of picoseconds, which are trillionths of a second. They tested multiple anesthetics, including xenon, which, as discussed earlier, is a noble gas that causes unconsciousness despite being chemically inert, as well as isoflurane and other commonly used general anesthetics. The results were striking and provided the first direct experimental evidence that anesthetics physically interact with microtubules in ways that affect their quantum optical properties. When anesthetics were added to the solution containing microtubules, the fluorescence decay was delayed by approximately 20% compared to microtubules without anesthetics. This 20% delay meant that the excited electronic states in tryptophan were persisting longer before emitting their fluorescence photons, indicating that the anesthetics were somehow stabilizing these excited states or altering the pathways by which energy dissipates in the protein. More detailed analysis revealed that the anesthetics were binding specifically to hydrophobic pockets within the tubulin proteins, exactly as Hameroff had predicted decades earlier. These hydrophobic pockets are formed by clusters of non-polar amino acids, including tryptophan, phenylalanine, and leucine, creating water excluding regions where the anesthetic molecules preferentially accumulate. Computational modeling confirmed that anesthetic binding to these sites causes subtle conformational changes in the tubulin protein that alter the electronic environment of nearby tryptophan residues. The altered electronic environment changes how quickly excited electrons can relax back to the ground state, which manifests as the observed delay in fluorescence decay. Critically, the magnitude of the fluorescence delay correlated with anesthetic potency. Xenon, which is one of the most potent anesthetics despite being chemically inert, produced a larger delay than weaker anesthetics. This correlation strongly suggests that the interaction between anesthetics and microtubules is not some random side effect, but is directly related to how anesthetics cause unconsciousness. If anesthetics caused unconsciousness by acting on synaptic receptors as traditional neuroscience suggests, there would be no particular reason for them to interact with microtubules in a way that correlates with anesthetic potency. But if consciousness involves quantum processes in microtubules, as Orko-R proposes, then anesthetics that more effectively disrupt these quantum processes should be more potent, which is exactly what the data showed. This represented a major validation of the Orko-R prediction that anesthetics work by interfering with quantum states in microtubules, rather than solely through effects on neuronal synapses. However, it is important to understand what this experiment did and did not prove. The fluorescence measurements demonstrated that anesthetics bind to microtubules and affect electronic dynamics in tryptophan residues, providing a clear biological link between anesthetics and microtubule function. What the 2023 experiments did not prove was that microtubules exhibit genuine quantum effects, like superposition or entanglement, under physiological conditions. The fluorescence delay could potentially be explained by classical changes in protein conformation and electronic structure without invoking quantum mechanics, showing that anesthetics affect tryptophan fluorescence in microtubules is consistent with quantum consciousness theories, but does not definitively prove them. To make that stronger claim, researchers would need to demonstrate quantum signatures like superradiance, quantum entanglement between multiple tubulin proteins, or quantum coherence lasting for physiologically relevant timescales. These are much more stringent tests because they require showing not just that electrons in tryptophan behave unusually, but that they exhibit behaviors that can only be explained by quantum mechanics and cannot be reproduced by any classical model. Nevertheless, the 2023 microtubule anesthetic experiments marked a crucial turning point because they established that the OR theory makes correct biological predictions. The theory said anesthetics should bind to specific sites in tubulin, that this binding should correlate with anesthetic potency, and that it should affect electronic properties of aromatic amino acids. All three predictions were confirmed. The findings also reignited interest in alternative mechanisms of anesthesia beyond the traditional synaptic receptor hypothesis. For decades, anesthesiology textbooks taught that general anesthetics work primarily by enhancing GABA receptors, which are inhibitory synaptic receptors that reduce neural activity when activated. According to this view, anesthetics flood the brain with inhibitory signals that prevent the widespread neural communication thought necessary for consciousness. But this explanation has always had problems. Different anesthetics have vastly different chemical structures ranging from simple noble gases to complex organic molecules, yet they all produce the same effect at roughly the same concentration when measured by their ability to dissolve in lipids, the Meyer-Overton correlation mentioned earlier. If anesthetics worked through specific receptor binding, you would expect molecules with different structures to have different potencies unrelated to their physical properties, like lipid solubility. The fact that lipid solubility predicts anesthetic potency better than chemical structure suggests that anesthetics work through physical effects on lipid membranes or hydrophobic protein regions rather than through specific chemical binding to receptor sites. Furthermore, some anesthetics do not enhance GABA receptors at all, and blocking GABA receptors does not always reverse anesthesia, indicating that the GABA mechanism cannot be the complete explanation. The microtubule hypothesis provides an alternative framework that can explain the Meyer-Overton correlation naturally. Anesthetics accumulate in hydrophobic pockets in tubulin proportional to their lipid solubility, and more accumulation causes greater disruption of quantum states in microtubules, leading to stronger anesthetic effects. This also explains why anesthetics affect organisms that lack nervous systems, like single-celled paramecia, because these organisms have microtubules but no neurons or synapses. When you expose paramecia to anesthetics, they stop swimming and become immobile, then recover normal function when the anesthetic is removed. suggesting that anesthetics disrupt some fundamental cellular process mediated by microtubules. The 2023 experiments therefore not only supported quantum consciousness theories, but also suggested a paradigm shift in how we understand anesthesia itself, with potential implications for developing better anesthetic drugs and understanding complications like post-operative cognitive dysfunction, where patients experience memory and thinking problems after surgery that may be related to anesthetic effects on microtubules in brain cells. Superradiance was first predicted by physicist Robert Dicker in 1954 as a peculiar phenomenon where a collection of atoms can emit light collectively in a coordinated burst that is far brighter and faster than the sum of individual atomic emissions. The phenomenon occurs when multiple atoms are arranged in a spatial configuration smaller than the wavelength of the light they emit, and when their electronic excitations become quantum, mechanically entangled, so that they act as a single giant atom rather than a collection of independent emitters. Under these conditions, when the atoms decay from their excited state back to the ground state, they emit photons in phase with each other, creating coherent constructive interference that produces an intense pulse of light. The intensity scales as the square of the number of atoms involved, so 10 atoms produce light 100 times brighter than a single atom, and 1,000 atoms produce light 1,000,000 times brighter. This quadratic scaling is the signature of quantum coherence and collective behavior, and it can only occur if the atoms maintain quantum entanglement during the emission process. Detecting superradiance in a system therefore provides direct evidence of quantum effects spanning multiple particles, making it an ideal experimental test for whether biological molecules can exhibit genuine quantum behavior under physiological conditions. In 2024, researchers conducted a groundbreaking experiment using large networks of tryptophan arranged in configurations designed to mimic the structure of microtubules. The experiment built on decades of quantum optics research and recent advances in molecular spectroscopy that made it possible to detect extremely faint and fast optical signals from biological samples. The researchers synthesized artificial structures containing hundreds to thousands of tryptophan molecules arranged in cylindrical lattices with geometry matching real microtubules. These synthetic microtubule analogues maintain the precise spacing and symmetry of tubulin proteins, but were simplified systems that could be studied under controlled conditions without the complexity of living cells. The key was creating structures where tryptophan residues were positioned at regular intervals around the cylinder surface, separated by distances of approximately 8 to 10 nanometers, which is the typical spacing in biological microtubules. This regular geometric arrangement is essential because it allows electronic excitations on different tryptophans to interact through their electromagnetic fields and potentially become entangled in collective quantum states. The experimental procedure involved exposing these tryptophan networks to pulses of ultraviolet light at wavelengths around 280 nanometers, which is the absorption peak for tryptophan's indole ring electrons. When the UV light hits the sample, it excites electrons from their ground state to higher energy orbitals, creating a population of excited tryptophan molecules across the network. In classical emission, each tryptophan would then independently decay back to the ground state over a time scale, determined by the intrinsic properties of the indole molecule, emitting fluorescence photons randomly with no particular phase relationship between different molecules. The total emitted light intensity would simply be the sum of contributions from all the individual tryptophans, scaling linearly with the number of molecules. But if quantum coherence develops across the network, the excited electrons on different tryptophans can become entangled through electromagnetic interactions, forming a collective quantum state. When this collective state decays, the emission becomes correlated across all the molecules, producing superradiant bursts where photons are emitted in phase, creating much brighter and faster light pulses than classical emission would allow. The researchers used sophisticated time-resolved spectroscopy equipment capable of measuring light intensity changes on picosecond timescales to detect these superradiant bursts. What they observed was extraordinary. The tryptophan networks produced intense, rapid pulses of ultraviolet emission with timing and intensity characteristics that could only be explained by superradiance. The emitted light was far brighter than expected, from classical independent emission showing the characteristic quadratic scaling with the number of tryptophans that is the signature of quantum collective effects. The emission also occurred much faster than classical fluorescence decay, with photons released in coordinated bursts rather than gradually over nanoseconds. Most importantly, these superradiant effects only occurred when the tryptophans were arranged in the symmetric, crystal-like networks that mimic real microtubule structure. When the researchers disrupted the geometric order by randomly spacing the tryptophans or by altering the cylindrical lattice structure, the superradiance disappeared and emission returned to classical independent decay. This demonstrates that the quantum coherence depends critically on the precise spatial organization of molecules, exactly as the Orch-OR theory predicts for biological microtubules. The 2024 discovery represents the first experimental evidence that microtubule structures can support quantum effects under conditions approaching biological relevance. Previous quantum effects in biological systems had been observed in photosynthesis, where quantum coherence helps energy transfer in plant light harvesting complexes, and in bird navigation, where quantum entanglement may help birds sense magnetic fields. But those phenomena occur in specialized proteins optimized by evolution for quantum effects, and they typically involve just a few molecules over very short distances and timescales. The microtubule superradiance is remarkable because it involves hundreds to thousands of molecules, coordinating quantum mechanically across distances of tens to hundreds of nanometers, which is enormous by quantum standards. Furthermore, while the initial experiments used simplified synthetic structures rather than real biological microtubules in living cells, the geometric requirements for superradiance match the actual structure of microtubules in neurons. Tubulin proteins in microtubules have tryptophan residues positioned at regular intervals with the precise spacing and symmetry needed to support collective quantum emission, suggesting that this capability exists in real brain tissue and is not just an artifact of artificial laboratory constructs. An additional crucial finding was that the quantum effects were sensitive to temperature and environmental perturbations, in ways predicted by quantum theory. When the researchers increased the temperature of the samples or added molecules that disrupt the ordered structure, the superradiance weakened or disappeared, indicating loss of quantum coherence. This temperature dependence confirms that the effect is genuinely quantum mechanical rather than some classical optical phenomenon that happens to produce similar signatures. At room temperature, which is close to body temperature, the superradiance was still detectable but weaker than at lower temperatures, showing that thermal motion does indeed cause decoherence, but does not completely destroy quantum effects as earlier critics had assumed. The fact that quantum coherence can survive even partially at physiological temperatures in these ordered molecular networks challenges the conventional wisdom that quantum effects require near absolute zero conditions. It suggests that biological systems have found ways to maintain quantum coherence despite thermal noise, potentially through the geometric organization and shielding effects of protein structures like microtubules. The implications of detecting superradiance in tryptophan networks extend far beyond just validating one prediction of the OR model. It demonstrates that the physical substrate proposed by Penrose and Hameroff, namely ordered arrays of aromatic amino acids in protein structures, can indeed support quantum phenomena under realistic conditions. This shifts the burden of proof. Skeptics can no longer dismiss quantum consciousness theories simply by asserting that quantum effects are impossible in biological tissue. The experimental data proves they are possible, at least in principle. The remaining questions are whether these quantum effects actually occur in living neurons, whether they last long enough and involve enough molecules to have computational significance, and most importantly, whether they have any causal role in generating consciousness. These are still open questions that require further investigation, but the 2024 breakthrough establishes the necessary foundation by showing that the physics permits quantum behavior in microtubule-like structures. It transforms quantum consciousness from a seemingly impossible speculation into a plausible hypothesis worthy of serious experimental investigation, marking a turning point in how the scientific community views the potential role of quantum mechanics in brain function and the nature of human awareness. Approximately 50 million people worldwide suffer from dementia, with Alzheimer's disease accounting for 60 to 70% of cases. One of the hallmark pathological features of Alzheimer's disease is the breakdown and disintegration of microtubules in neurons, caused by abnormal phosphorylation of tau proteins that normally stabilize microtubules but become dysfunctional in the disease. When tau proteins are hyperphosphorylated, they detach from microtubules and aggregate into tangled filaments, causing the microtubule networks to collapse. This microtubule breakdown correlates strongly with cognitive decline. The more microtubules are disrupted, the worse the memory loss and cognitive impairment. Traditional neuroscience explains this correlation by noting that microtubules are essential for transporting neurotransmitters and other molecules within neurons, so their breakdown impairs synaptic function. But if microtubules are actually the quantum substrate of consciousness, as the OR theory proposes, then their breakdown would directly destroy the physical basis of conscious experience and memory formation, providing a much more fundamental explanation for why Alzheimer's disease causes dementia. This connection between microtubule disruption and consciousness loss opens entirely new therapeutic possibilities. Drugs that stabilize microtubules or restore their quantum coherent properties could potentially treat or prevent cognitive decline by preserving the quantum substrate of consciousness, rather than merely supporting molecular transport. The discovery that microtubules can support quantum effects does not prove that consciousness is quantum, and this distinction is crucial for understanding what has and has not been established by recent experiments. The 2024 super radiance experiments demonstrated that tryptophan networks with microtubule-like geometry can exhibit quantum coherence and collective emission. The 2023 anesthetic experiments showed that anesthetics bind to microtubules and affect their optical properties in ways that correlate with anesthetic potency. Together, these findings establish that the biological mechanism proposed by Penrose and Hameroff is physically plausible. Microtubules can support quantum phenomena, and disrupting these phenomena correlates with loss of consciousness caused by anesthetics. However, plausibility is not proof. To definitively establish that quantum processes in microtubules generate consciousness, researchers would need to show several additional things. First, that quantum coherence actually occurs in microtubules within living neurons in intact brains, not just in isolated artificial structures. Second, that this quantum coherence lasts long enough and involves enough molecules to perform computationally significant operations. Third, that disrupting quantum coherence in microtubules causes specific changes in conscious experience that cannot be explained by classical effects on neural activity. And fourth, that the information processing performed by quantum microtubule states actually contributes to cognitive functions like perception, memory, or decision-making. Some of these requirements are beginning to be addressed by cutting-edge experiments. In 2022 and 2023, researchers using novel magnetic resonance imaging techniques reported evidence of quantum entanglement in the living human brain that correlates with consciousness and working memory performance. The experiments used an unconventional MRI protocol designed to detect signals that could only arise from quantum entangled states involving nuclear spins in water molecules. The researchers observed signals that mimicked heartbeat-evoked potentials, but had characteristics suggesting they originated from quantum-entangled brain states rather than classical neural activity. Most significantly, the strength of these putative quantum signals correlated with how well subjects performed on working memory tests, and the signals disappeared when subjects were asleep compared to when they were awake. If these findings are confirmed by independent replication, they would provide direct evidence that quantum states exist in the conscious human brain and play a functional role in cognition, strongly supporting quantum consciousness theories. However, these experiments are technically challenging and controversial, and many scientists remain skeptical about whether the observed MRI signals genuinely indicate quantum entanglement or reflect some classical artifact that happens to produce similar signatures. Additional experimental evidence comes from studies of microtubule resonances in living neurons. Researchers stimulated cultured neurons and detected resonance frequencies in the megahertz to gigahertz range that appeared to originate from microtubules and could span across multiple neurons, with the resonance state influencing membrane voltage and potentially coordinating neural activity. These observations suggest that microtubules act as oscillators that can become synchronized across cellular and network scales, creating conditions favorable for quantum coherence to emerge and persist. The axon initial segment, which is the region where action potentials are generated in neurons, has been identified as a particularly promising location for quantum effects because it contains highly organized fascicles of microtubules with unique structural properties that could support spintronic oscillations and long-lived quantum coherence. According to recent theoretical work, the characteristic features of the axon initial segment, including the high density of microtubules, specific arrangement of microtubule-associated proteins, and particular electromagnetic environment, create optimal conditions for spontaneous quantum coherence to emerge in microtubule populations. This localization of quantum effects to specific neuronal compartments could help explain how quantum and classical processes integrate in brain function without requiring the entire brain to behave quantum mechanically. The potential applications of quantum consciousness research extend far beyond fundamental neuroscience into practical medical and technological domains. Understanding how anesthetics work at the quantum level could lead to development of safer anesthetic drugs with fewer side effects and better targeting of the specific quantum processes that cause unconsciousness rather than broadly suppressing neural activity. Postoperative cognitive dysfunction, where patients experience persistent memory and thinking problems after surgery, affects millions of people annually and may result from anesthetic damage to microtubules in brain cells. If this mechanism is confirmed, protective strategies could be developed to minimize microtubule disruption during anesthesia. For neurodegenerative diseases like Alzheimer's and related dementias, therapies aimed at preserving or restoring microtubule quantum coherence could address the root cause of cognitive decline rather than just treating symptoms. Small molecules that stabilize microtubule structure or enhance quantum coherence properties might prevent or reverse the consciousness loss associated with tau pathology and microtubule breakdown. The implications for artificial intelligence and machine consciousness are equally profound. If consciousness requires quantum processes that cannot be replicated by classical computation, then current approaches to artificial intelligence, which are based entirely on classical computing architectures, could never produce genuinely conscious machines, no matter how sophisticated the algorithms become. This would mean that the human mind has capabilities fundamentally beyond what silicon-based computers can achieve, preserving an essential distinction between human consciousness and artificial intelligence. Alternatively, if we could engineer artificial systems that support quantum coherence in structures analogous to microtubules, we might be able to create genuinely conscious artificial entities. This possibility is being explored theoretically through proposals for consciousness transfer technology based on mapping neural network states onto microtubule quantum states in artificial substrates. While such technologies remain highly speculative and face enormous practical and ethical challenges, the quantum consciousness framework at least provides a conceptual basis for thinking about how consciousness could potentially be transferred or replicated in non-biological systems. More experiments are urgently needed to distinguish between competing hypotheses and rule out alternative explanations for the observed phenomena. The superradiance detected in tryptophan networks could potentially arise from classical optical effects or contamination by other fluorescent molecules, so careful control experiments are essential to confirm that the effect is genuinely quantum mechanical and originates from the tryptophan in microtubule structures. The correlation between anesthetic binding and fluorescence changes needs to be tested with a wider range of anesthetic and non-anesthetic compounds to ensure the relationship is specific rather than a general effect of hydrophobic molecules binding to proteins. The MRI signals interpreted as evidence of quantum entanglement in human brains require independent replication and more rigorous theoretical analysis to rule out classical explanations. Future studies should also investigate whether disrupting microtubules, specifically using drugs or genetic manipulations that affect microtubule stability without broadly affecting other cellular processes, produces predictable changes in conscious experience that match theoretical predictions. If quantum processes in microtubules really generate consciousness, then interventions that enhance quantum coherence should enhance conscious awareness, while interventions that disrupt coherence should diminish consciousness in specific ways. Testing these predictions will require creative experimental designs combining molecular biology, quantum measurement techniques, neuroscience methods, and careful assessment of subjective conscious experience. The convergence of evidence from multiple independent approaches would build a compelling case that quantum mechanics plays a genuine causal role in human consciousness, transforming our understanding of mind and brain.